Dear parents, so as you related, Miss West causes sickle cell anemia and suffers a lot with the disease. However, Mr. West don't know about his DNA, if he's a carrier of this illness and if past generations of his family has it. Therefore, you are consulting us to find out what are the probabilities of your son being born with sickle cell anemia and if you must be concerned about this topic.
So, it is explainable that you are afraid of your baby's future, since the sickle cell anemia is an inheritance disease and it has a lot of consequences in the lives of the babies, like intense pain crisis, prone to infection, and other terrible things that commit these people. As one of you has it, the probability increases a lot, because if the father also has a recessive trait, there is a huge chance that your son be a carrier and manifest the disease. Thus, your preoccupation is not in vain, but with genetic counseling, we can predict the possibilities and try to guarantee a better future.
Now, me and the other doctors are going to start explaining about the anemia disease, talking a little bit about the DNA structure and how everything works so we can show the probabilities of your baby.
Firstly, I will explain what a gene are. So, genes are a section of the DNA that is responsible for building certain proteins and RNA molecules. Humans have about 20,000 or 25,000 genes in their bodies. It gives instructions for specific traits. Additionally, this section is passed from parents to the offspring.
Now, I'm going to talk about allele, refer to two or more different forms of genes located at the same position on homologous chromosomes and controlling the same trait. In simple terms, alleles can be thought of as different versions of the same gene. For example, in Mendel-P experiment, the gene controlling flower color had two alleles, one that protects purple flowers and the other that protects white flower. In a diploid organism, such as a human, each trait is determined by two alleles.
Now, the last thing that I'm going to explain is about chromosomes. There are thin structures that are made of proteins and a single DNA molecule whose function is to transport genomic information from one cell to another. In eukaryotes, the chromosomes are located inside the nucleus of a cell. Humans have 22 pairs of numbered chromosomes and one pair of sex chromosomes, that's called XX and XY. So in total, each cells have 46 chromosomes. Each pair contains two chromosomes, one from the mother and one from the father, and this happens with all the chromosomes. So half of them is from each of the baby's parents. Now in this first image, we can see how a chromosome looks like on a representation that came from a drawing. And we can see that each one of these legs, it's representing one chromosome from each parent. And when they are together, they are a homologous chromosome. Now in this other picture, we can see the 23 pairs of chromosomes, being the ones that are separated, the sex chromosomes, and the others, the autosomes. And we can see that all of them are different because of their alleles and their structures that represent different things.
You understand what is a gene, a chromosome, and an allele. So, I will explain a little bit about dominant and recessive allele that is important to determine and predict the possibilities of the outcome. So, alleles can be dominant or recessive. If it is dominant, it means that the trait just needs one copy of the allele to appear in the physical appearance, and literally domines over the other gene, the recessive one. Nonetheless, if it is a recessive allele, it needs two copies to appear in the physical traits of an organism. To comprehend the situation in genetic matters, it's important to know what is genotype, phenotype, and their difference. So, genotype refers to the genetic material. It is a term that refers to two alleles that a person has. So, the possibilities could be heterozygous, meaning that the person possesses one dominant allele and one recessive allele, but the dominant allele is visible on the physical characteristics. This is represented by big B, little b. Let's go to the second option. There is also the option of being homozygous dominant. In other words, the gene possesses both dominant alleles, represented by big B, big B. Furthermore, there is a third option, homozygous recessive, meaning that the gene possesses two recessive alleles, written as little b, little b. Phenotype are the physical traits. In other words, are the characteristics that are observed. For example, hair color, skin texture, eye format, body shape, and blood type. Furthermore, it is influenced by genotypes, but also for the environments that the person or the organism lives. This picture summarizes and explains what is genotype and phenotype and also give examples, so you could understand in a better way. Since we know about these rules, it is easy to explain traits and inheritance from parents to the children. Thus, when meiosis occurs, a haploid cell is formed, more specifically a sperm or an egg. These gametes contains only one allele copy. So, at the moment where fertilization is initiated, the allele for the mother and the father get together, creating the genetic materials of the baby.
Now I'm going to talk about a useful tool that we doctors and everyone that wants to predict something about the DNA genes and the alleles use for this prediction. This is called the Punnett square. This also includes predicts crosses between plants, animals, and even humans. Normally, Punnett squares are used with Mendelian inheritance. Mendel was the scientist who helped pave the way for genetics and is known today as the father of genetics, giving us a basic and fundamental understanding of how traits work. Now here we have an example. These two numbers above are normally the male's alleles, and these two numbers in the left are normally the female alleles. We can see that in this example, the mother and the father, they are heterozygous, so any one of them have the trait. After, we will match both of them. So if we match these two that are in this square, we're going to get the big B and big B. So we have a dominant trait. Now, if we match this big B with this little b, we're going to get an heterozygous result. The same thing when we match this big B with this little b, that we're going also to get an heterozygous result. And now if we match this little b with this little b, we're going to get a recessive result. If we observe, we can define that we have 75% of chance of the baby don't having the traits and 25% of chance of the baby having the trait. But we also have 50% of chance that the baby will be a carrier and only 25% that the baby will have nothing related to the disease. Here we have the real problem. We know that you, the mother, is homozygous recessive, so you are little b. But we know that the father don't know which genetic information he have. But since he don't have the disease, we know that one of his alleles will be a big B.
Let's apply these concepts in the real life case. So the first case is if your husband is a heterozygous. So it means that he's a carrier of this disease. So we do a Punnett square and we calculate. In this calculation, we will get big A little a here, big A little a, and little a. Therefore, it has 50 of chance to have the disease. Also, has a 50% of your son be a carrier or a heterozygous, and 50% of the son be a homozygous recessive. So we do the same thing in case 2. So the case 2 is if the father is a homozygous dominant, big A, little a. So all the results possible, it's heterozygous, meaning that has 0 of chance of your son have the disease.
Now I'm going to talk a little bit about the DNA. So DNA, also known as deoxyribonucleic acid, is a molecule that contains genetic instructions for living organisms to develop, function, and reproduce. All living things have DNA in their cells. It is a set of instructions for creating the proteins that make your body work. 2 strands of DNA together form a double helix. You can think also about a spiral stair if you think about the double helix. Our DNA is made of sugar and phosphates that makes the side of the DNA, and from bases that makes the middle part. The bases from our DNA is known as adenine, or you can say that is letter A, cytosine, that is letter C, guanine, that is letter G, and thymine, that is letter T. You can think of DNA as a vast library of instructions manual. It uses its own language to write the story of you that your cell can read, defining what you will look like.
Sickle cell disease is a disorder of blood. It alters the typical form and flexibility of red blood cells. Healthy red blood cells can pass easily through the blood vessels and distribute oxygen efficiently throughout the body. People with sickle cell disease have red blood cells that become rigid and sickle shape. This sickle shape cells, specific cells of all blood cells, hinder blood flow, break more easily, and contribute to debilitating pain, fatigue, and other serious health. This disorder is primarily the result of a change to the HBB gene, which is responsible for instructions for one of the proteins in red blood cells. Sickle cell disease is formed when normal hemoglobin is altered. The sickle cells form when the red blood cells contain primarily the abnormal type, HBS. When blood oxygen levels are low, HBS proteins bind together, and the red blood cells become sickle-shaped.
Sickle cell disease is passed down in families through the recessive gene. This means a person has to get one sickle cell gene from each parent to have the disease. If someone gets only one sickle cell gene and one normal gene, they are called the carriers or have the sickle cell trait. Carriers usually do not get sick, but they can pass the gene to their children.
In conclusion, sickle cell disease is a serious inheritance illness caused by the change in the HBB gene. It has changed the shape of the red blood cells and can cause many health problems. Here in this image, we can analyze and compare the blood cells, one with sickle cell anemia and the other without. So the shape is different and it can cause many problems. Let's go to the conclusion of the situation. As we observe in the probabilities calculated in the Punnett square, the baby has 50% of being born with this disease if your husband have a recessive allele. However, if he is a homozygous dominant, this chance reduced to 0%. Therefore, I advise Mrs. Smith to have a genetic test done, so he can be sure if there is any probability of the baby having this illness. I also recommend that he try to find any appearance case in his family. If Mr. Smith has a recessive allele, I suggest that you reflect on the life of these children, and if it's truly a dream that needs to be complete, accept that perhaps the child will need additional care and lead a more complicated life.
For our student reflection, we came with some conclusions. Genetic counseling using scientific knowledge to improve the quality of life and predict what is the probability to an outcome have certain predisposition to diseases or other health problems. Another thing is that genetics counseling has a power to affect not only the scientific aspects, but also in the society ethical and emotional. For example, if a family dreamed to have a baby, and according to the genetics, it will happen healthily, even with a past disease case in the family, their emotions would be affected and they would get happy as the entire community around, impacting in a society and emotion aspects. Furthermore, in the ethical aspects, genetics counseling could help to determine if a couple should or not have a baby considered that he will suffer. So ethically, this partner should avoid having kids.